Hello, AP Bio Bruins, and today we'll talk about active transport. Previously we talked about passive transport. Now we'll talk about active, and when we say active, it means that we're going to use energy and we'll go against the gradient. Now there's two types of active transport. That's called endocytosis and exocytosis, and that's basically the cell engulfing and the cell releasing. And then we'll talk about different types of active transport like sodium potassium pump, hydrogen pump. Now, just a quick review. If you remember, diffusion and facilitated diffusion are part of passive transport. And when we say passive transport, remember, uses no energy and it goes along the gradient. And if you remember what that word gradient means, differences, and here we're talking about differences in concentration. So it's always going from high concentration to low concentration. So if you remember, diffusion is again high concentration to low. And what are some things that use simple diffusion? And if you remember, the cell membrane's made out of phospholipids, so small non-polar particles will just simply pass through. And examples are lipids, CO2, O2. Facilitated diffusion requires a helper protein, and this protein acts like a channel or a gate. And things like water, so these particles could be something charged or large, and particles like water, like we have aquaporin as an example. These channels could be for particular ions. So large and charged particles, and also importantly sugar. Now, active transport establish a concentration gradient, meaning you want something higher concentration in one area and lower concentration in another, and it usually uses energy in form of ATP or ion concentration. So there's two types. So active transport uses energy and it goes against the gradient. So when we say against a gradient, we're talking about low to high. Kind of think of going up the hill. So when you go up the hill, you're going against the gradient and you need energy to go up the hill. So that's active transport. Okay. So what kind of things transport by active transport? And that's usually ions to establish that electrochemical gradient that you need, especially like in your nervous system. Another big ion that is hydrogen ions, and we'll see that in cellular respiration and photosynthesis. And another big example is sodium potassium pump. Okay. Now, a good example of active transport is a sodium potassium pump, and the sodium potassium pump is basically a proton pump, and basically you have 3 sodium ions going out, and you have 2 potassium ions going in. So what happens is, first, this protein has a high affinity toward sodium, so the sodium ions bind, and ATPs get hydrolyzed and phosphorylates this protein. And whenever you phosphorylate this protein, you see a conformational change, and that conformational change causes the sodium ions to be released. So this protein no longer has that high affinity for it. Then the sodium potassium is expelled and has a high affinity for potassium ions, and these potassium ions are bind to the protein, and we see the phosphorylate dephosphorylates, and when it dephosphorylates, it has a conformational change, and this conformational change causes the release of potassium in the cell. So if you want to see the animation, I'll put a PowerPoint and you can see the animation about sodium potassium pump. So another good example of active transport is called a proton pump. So here in the proton pump, you're using the hydrolysis of ATP to create a hydrogen concentration gradient. So here in one side of the membrane, you have a high concentration of hydrogen, and the other side you have a low concentration gradient. Now here's an example where we're using the energy of the high hydrogen concentration, and we establish this gradient by using this proton pump, and then later on this high hydrogen concentration is going to diffuse through this protein, and while this is diffusing, that energy is used to co-transport sucrose, a sugar. So here we're not using ATP, but it's using the energy from hydrogen being diffused from high concentration to low concentration. So here's an example of coupling where this required energy, and then this released energy. Now another example of active transports is where the cell membrane will either surround a particle and engulf it, and that's endocytosis. So remember, the word endo means in, and cytosis means cell. So here the membrane will surround that particle and form a vesicle around it. So you're basically separating these particles from the rest of the cell, and basically release enzymes in here, and they could digest this. So here's an example of an amoeba engulfing a bacteria, and this will be isolated in a vesicle, and where enzymes will digest it. Now phagocytosis, basically think of it as cell eating, and pinocytosis, think of cell drinking. So the particles are smaller and liquid. So again, the cell membrane will surround it, and you'll see a vesicle around it, and again enzymes could be released to break it down. Now in exocytosis, very similar to endocytosis, and the word exo means to release out or exit. So here the cell membrane, you have your packaged item, and this could be a protein or enzyme or a hormone that is separated from the other parts of the cell, and then emerges into the outer membrane, and then releases the particles in here, and that's exocytosis. So again, if you want to see the animations, go to the PowerPoint on Blackboard.